Hey y'all, I'm Steph Billman. I'm one of the IDAS ATP trainees, and I'm very excited today to introduce Dr. Emmy Bruns. Dr. Bruns is from the University of Maryland today. She did her PhD at the University of Minnesota, studying constraints to oak crown rust virulent evolution. She then did a postdoc at UVA studying disease dynamics at species range limits in the Alps. She is now an assistant professor at the University of Maryland in the Department of Biology, studying ecological and evolutionary dynamics of infectious diseases in natural populations, understanding impacts of infectious disease on host abundance and distribution, and how these ecological effects intersect with evolution of host and pathogen transmission and resistance. Thank you for coming. We're excited for your talk.
Thanks. Well, thank you so much for the invitation. It's really exciting to be at Emory, and I just had a great time hearing about all of the different stuff you guys are doing. It's exciting to be around so many disease people. So today, usually when I give a talk, okay, hopefully it's gonna work now. Classic, right? There we go. Usually, when I start giving a talk, I spend the first slide just pumping everybody up about how crazy cool parasites are, right? And usually I do that by pointing to one of my favorite studies from Armand Curris's lab and Kevin Lafferty in Southern California, where they just describe the abundance of parasites in a single salt marsh. And they say, okay, they counted up and they said the total biomass of parasites in the salt marsh was actually equivalent to the biomass of birds. So this is mostly like trematode parasites. So just astounding, right? And we think globally, it's been estimated that 40 to 50% of all species are parasites. This is probably an underestimate. I'm sure people are thinking about viruses with this, with parasites we can see probably here. So they're just everywhere. And so as an ecologist, it's an exciting thing to study, right? You guys are all disease people, so I probably don't need to pump you up about disease, but what I might have to do is get you excited about plants, because I'm going to talk about plants today. So plant diseases are really cool, and they're like the stars. So basically, the more you look, the more there are. And one of the things that got me into it when I first started was I took a plant disease ecology class, and the professor just took us for a hike in the woods. And he said, find disease. And you start looking, and then it's everywhere. And you sort of wonder, and the question that you end up wondering is, well, how the heck are the plants making it, right? The world is green, and their plants are under constant, they have to sort of stand and fight. They can't get up, they can't run away. They're being bombarded with pathogens, fungal pathogens, bacterial pathogens, viruses even, all this stuff. How do they make it? And so sort of the fundamental question that I think I can now say my lab is interested in is we're really interested in the ecological and evolutionary factors that lead to host pathogen coexistence in natural populations. So how can you have this exist? How can the world still be green? And sort of what are those two dynamic pressures on the ecological side? How do fluctuating population numbers maintain? And then we know that there's an evolutionary component to this, right? Things are not staying the same. They're using evolution to try and get ahead of their antagonists. So that's really the work that we're interested in. And today, I'll tell you, and of course, I say this very broadly, but most of my work is actually just thinking about one particular parasite that I've host-pathogen interaction that I've worked on for about the last 10 years. And so I'll just introduce it to you right now. It is, among all of the plant diseases out there, in my opinion, one of the coolest. It's called anther smut disease. It's caused by a basidiomycete fungus in the genus Microbacterium. And here's some teleospores of it. And what it does is it infects flowers. It's a healthy flower. So these spores land on the plant, not necessarily on the flower, they could land on the leaves. They'll germinate and infect the plant, and they cause a completely asymptomatic infection. So they grow in between the cells of the plants and the meristome, that's the growing tissue of the plant, and they cause no symptoms. So there's no mortality. This plant's healthy. You can't tell an infected plant from an unaffected plant. But the next year when the plant flowers, it's the fungus sporulates in the anthers of the plant. So this would be the pollen-producing areas of the plant. And so this plant is sterilized, so it's not producing any male components, not producing any pollen. It also sterilizes the ovary, so if the plant's a hermaphrodite or a female, it can't make seeds anymore. And it leads to a perennial disease. So once the plant's infected, that's it. It's not going to recover. Every year, it's producing more, just producing these smutty, sterile flowers that are then pushing disease out into the population. And the other cool thing about this is you can look at this and you might be able to imagine how transmission works. So this still looks like a flower, kind of looks like a pollen color morph. And so it still gets visited by butterflies. And so, and bees and hoverflies and even like little small flies. So pollinators still visit it. And when they move to healthy flowers, they can actually transmit the disease. And so it's a nice system to study. One of the things you can see is you just see it in nature. And so when we go out and we're trying to look and see how much disease is there, we can just count very easily disease plants from healthy plants. And because it doesn't kill the plant, we can get good estimates of prevalence because the disease isn't removing infected individuals from the population. So that's the species that I've gotten really excited about and trying to understand sort of these questions of how does the host and pathogen persist in nature and what are these dynamics. And so today I'm going to tell you two stories from some of this work. The first one is going to focus on this question of persistence. And I want to tell you how we've discovered that the disease is actually persisting through some cryptic modes of transmission in nature. And then I want to shift and spend most of the talk talking about resistance evolution with a focus on evolutionary dynamics of age-specific resistance. So that's sort of the talk. And then I thought I would give you the background to it because I was asked to do this a little bit, which is, so I got into working on this system. It's kind of a random system to work on. And I was basically finishing up a PhD, as Steph said, at University of Minnesota, working on this not as attractive fungal pathogen, a rust fungus, very agriculturally important one, looking at trade-offs with infectivity and a crop pathogen. And this was sort of my field study when I would go out and just go to North Dakota and like go into these big fields and collect leaves. And it was nice, but I was really wanting to do something back in natural systems. And so I got, there was a job ad floating around for a postdoc position with this guy, Janos Antonovics, and his buddy Michael Hood, and they just got this grant to look at anther smut disease transmission and species range limits in the Alps. And so I didn't think I was like ready to finish my PhD, but there's like this. And so I applied and I wasn't even quite ready to finish. And Janos said, well, you can take another, 5 months or something to finish, or 6 months, but you have to come out to the field with me this summer and help and motivate. And so this is basically why I work on this system now, because this is the place he took me. So this is in northern Italy, up by the border with France. And this is, if you were to go over this mountain here, Marguerite, that would be France on the backside here. And this is the field station that we work at. It's, I can't even say this with a straight face. It's beautiful. It's a refugio, and so you have to hike up to it and bring your equipment. But then when you're there, it's Italian, so there's really good food. There's espresso. There's wine in the evenings, and they actually have a botanical station that they've built out now, and we've got space there where we can grow plants actually up at the botanical station and use them for experiments. And the really cool thing that was so exciting about this actually, though, is that when we went there. This whole meadow for about 2-3 weeks in July turns pink with these flowers. So it's this incredibly abundant population, and it's just hammered with disease. So 43% of these plants are infected with this incurable sterilizing disease, right? And so this was such an exciting system to work on because it immediately asked the question, will the disease actually drive the population to extinction, right? Here we've got a disease that's sterilizing. We know that sterilizing diseases have larger impacts on population size and have a higher capacity to drive populations to extinction than mortality-inducing diseases because they're not killing their host. And we also think because it's pollinator transmitted, the dynamics would be more frequency dependent. And so it could survive even as the host population starts to decline, the pathogen won't drop past that threshold density. And so this was the question that sort of first took me when I started as a postdoc. And there was some additional data that really suggested this was probably occurring. So we had some transects that Janos and Mike had started back before my time, where they'd been monitoring the population size and the prevalence of disease. And what they found in both these transects, you can see, is that the population size, as the dashed line, is just tanking, but that the frequency of disease shown here on the y-axis or on the right axis is staying pretty rock solid at 40%. And so this really looked like disease-driven extinction. But of course we can't, yeah.
Is there some documented time at which like the anther smut was actually introduced into the system, or?
So if there is, we don't know, but it's actually I neglected to say, it's a naturally occurring pathogen of these carnations. And actually there's a whole bunch of individual anther smut species on other species of carnations. There's a whole species complex, and it's a long co-evolutionary history. So we think that they've co-evolved together for a long time. So in this particular meadow, we don't know when it first came in. It's a long-lived host too, so the epidemic is moving a little slower than, which is something we should have paid attention to at the beginning, actually. So what we thought, well, is it's not enough just to look at these patterns. You also want to like generate a model, right, and actually understand transmission. So we did a mark recapture study, which is easy to do with plants. You can tag them, they're still there. Giannis taught me that the phrase is plants stand and wait to be counted, and it's very true. And so we use metal detectors. We put little wires around. You go into the metal detector, you find the plant, and then you know a couple of really useful things. You know what the mortality rate is, and you know how many healthy plants get infected. And so we tagged a whole bunch of healthy plants. We monitored for 6 years to see how many got infected. And then we took all that in and we built a model. And here we can build actually a pretty simple, we don't even have to do an SIR model because there's no recovery. So you just build an SI model, and the only complication is that the plants don't flower every year. So sometimes they take a year off and they're vegetative. And so we have to, but we know from the data how often that transition occurs. And then what we did was we tried to fit the result. We simulated that model. We tried to fit it to the data that we saw in the field to see if we could model the transmission. And it turns out that we did a really crummy job. So here's the observed data here, and this is what our model predicts. So our model predicts that the disease should decline, and in fact, we see the disease maintain. When we first started doing this, I didn't know it was going to, I actually thought it was like a modeling problem on my end, because this was the very first transmission model I'd really done. But finally, I came around to the fact that it was actually the biology that we had wrong. And basically, it was just that our observed transmission rate was far too low to sustain the epidemic. So the transmission rate we observed from these marked plants, only 4% of the plants became infected over the whole 6 years. So it's really, really low. And interestingly, we know it's not limited by spore dispersal. So you can just go and collect healthy flowers, bring them back, wash them off in a test tube and count the spores. 80% of the flowers have spores all over them. So it's not an exposure issue. They're all getting exposed. And it was even more frustratingly we couldn't like say it was a susceptibility issue, because we can collect seeds straight from the field, grow them in petri dishes, infect them, and they were coming up 80% infected in the lab. So we didn't know what was going on. And we probably still wouldn't have known. So here's some more data that this is all seedling infections, and they keep getting highly infected depending on what you do. But then I was trying another experiment, and I actually cloned up some adult plants and tried to infect them. And all of a sudden, I couldn't do it. I got really, really low, actually got 4% infection rates on the adults. So we did this again with a controlled experiment where we had seedlings and adults. And lo and behold, you have this really strong age effect. So basically these seedlings are incredibly susceptible to the disease, and the adults are really resistant. And if you remember in that marked plant study, we had flowering plants because we thought all the transmission was occurring through pollinators. And so we weren't getting the transmission. We weren't really capturing the full transmission rate in the population. So the question, of course, is like, well, how do seedlings get infected with a disease that's being moved around by pollinators? Because these seedlings are little tiny things on the ground that you have butterflies like landing down in them. And so what we hypothesized was just that there might be a component of aerial dispersal. So this isn't like, it doesn't look like a wind dispersed pathogen, but maybe it has some level of spores that are falling down around the ground, and that could actually contact plants of all ages, including seedlings. And so we tested for this in this really high-tech way where we put out strips of double-sided tape, transparency paper out in the field, and did transects around heavily diseased plants. And sure enough, you can find spores in this spore shadow. It is a really limited distribution. So even about almost a meter out, you're still finding spores, but you can see that most of the deposition is quite, within 15, 25 centimeters. But keep in mind, these plants are really dense in the field. And so you've got lots of diseased plants. So actually the total amount of spore danger zones for seedlings could be quite high in these heavily diseased populations. So then we took our new information, I'm skimming through a lot of work here, but and we basically built the model out a little bit more and said, okay, well, what if we also have a juvenile class that can, this should be a dash line that's produced, by births, and this could have we assumed that there was frequency-dependent vector transmission between flowering plants and density-dependent transmission to juveniles and to vegetative plants. And then we also put in the susceptibility measures. So we said, okay, and assume that juveniles are on order. I mean, this was like, they were like 20% more, or 20 times, sorry, more susceptible than the adults. And so when we put all this in, basically what we found is that this mixed transmission mode, mixed vector and aerial transmission actually starts to explain the observed patterns of disease that we see in nature. So this model, I hope you can agree, looks a little bit better than the previous one. So we just hadn't accounted for transmission. So it turns out that in anthracite, there's 2 transmission modes. And one I use the phrase cryptic because it's so obvious. You can like watch a pollinator move, and then everything about the natural history suggests that that's how it's getting around. And this mode is sort of very inefficient, right? Spores are being scattered, and the chance of contact in the seedling is low, but the payoff is big because they're really susceptible. And more than that, we then use this model to basically ask, well, how important is aerial transmission for disease persistence and for host persistence? And so within the model, when we looked there, we found that in all those simulations we ran, about 70 to 80% of the total transmission events are actually to the seedling stage. And we've also found that disease just can't persist without seedling transmission. So in both cases, our model predicts that over time, the total amount of disease will decrease in this population. We don't think we can maintain it at 40% for that much longer into the future because basically the adults are so resistant, there's not very much going on. But without any seedling transmission at all, we would rapidly lose the disease. So we went from basically the scenario of thinking, okay, the disease is going to drive the host to extinction, to be like, okay, yeah, the host is fine, but how is the disease going to persist? And it turns out that on the one hand, adult resistance is allowing the host to persist, but seedling susceptibility is really what's allowing the disease to persist in these dense populations. And I wanted to add this because I know this is the disease across scales bit. So you might ask, well, do the pollinators matter at all? Are they just, are we just fooled because they're like interesting and fun to watch, but maybe they're not doing anything? Well, so for this, what we had done at the same time is for another project, we've been looking sort of across scales here, and this is the whole sort of Western Alps. And each one of these dots is a population that we sampled. So we were doing these elevational transects, and we ended up sampling 59 transects and mapped 700 host populations, not just of this species, but other ones as well, but mostly this one. And what we found is basically that in all cases, you can map the host range sort of in, that's shown in green. I don't have that individual data points on here. And this is a high elevation species. So it's really abundant at high elevations, and it has this sort of lower range margin around 1,500 meters, where the population sizes get low and sort of more scattered. But we find a lot of disease, even right up to the range margin and in those small marginal populations. And again, we wouldn't expect a density dependent disease. If transmission was purely aerial into seedlings, we really shouldn't, we should expect a disease-free halo. We shouldn't expect that the disease could persist through density dependent transmission alone. So we know that at the broad spatial scale, pollinators have to be incredibly important at sort of spreading the disease out to new populations and maintaining it in these low density populations. And so what we've now, I've sort of through all this, I've just generated a lot more respect for this fungus. It's really hit on this awesome transmission strategy. It's using both modes of transmission to maintain itself. And so at the vector transmission level, it's able to achieve this sort of widespread distribution. It's rare that we find populations that don't have at least a little bit of disease, and it can persist in these small marginal populations. But within these dense populations, aerial transmission allows it to exploit this susceptible juvenile stage. And this is actually a key trait for pathogen persistence in here. So without these susceptible seedlings and the ability to disperse, then the pathogen would rapidly go extinct as hosts evolve resistance. So anyways, we think this is a cool system for looking at evolution of transmission modes. And we have a couple of other things we're doing on that, but I'm gonna leave the transmission mode for a bit. And I wanted to spend the rest of the time talking about some work that's very related, that was all postdocs type stuff. This is the work that my lab has really been doing now, which is that at the end of this, I got really fascinated with this question, well, why the heck are the seedlings so susceptible, right? This doesn't seem like a good idea, because, if a seedling gets infected, then the very first flower it makes is smutty and sterile, and it's not going to reproduce its entire life. So it gets zero fitness, right? Whereas an adult, maybe it gets infected after it's had a few seasons of flowering, and so it's going to be less of a fitness cost. And I should just say before I start, this isn't just an anthracite problem. This is terrible, like to show a huge table. But what I wanted to do is convince you that, in fact, these are all different studies we've found in invertebrates, and crop plants, where people have gone in and done inoculation experiments. This is all old vet stuff where they were doing like guinea pigs at different ages, and showed basically that juvenile susceptibility is really a widespread phenomenon. So we know that in a lot of cases, young individuals are more susceptible to disease than older. When you get far on the other end, you can start to talk about, immunosenescence and that kind of thing. But here we're just thinking about sort of the early life to midlife to reproduction kind of a situation. And so this is really common. So it's not just an anther smut problem. And, I showed you an anther smut, it was epidemiologically important. It's epidemiologically important in lots of places. In humans, we know that children bear the brunt of a lot of the real hard work of transmitting our diseases for us, in wildlife, there's some nice observations showing that transmission of viruses can be linked to these birth pulses, right? Where you have in bats huge populations where all the pups are born at one time, and you get higher levels of transmission and potentially even spillover risk. And in plants, there's whole groups of diseases that just target seedling stages, damping off diseases. And the plants are completely resistant to them after they get a little bit older. But these diseases can actually be really important for even thinking about biodiversity. So James and Connell, plant ecologists out there, maybe none, but we think that these seedling diseases can be important in terms of thinking about sort of forest biodiversity. So that's my plug on why I think this is an interesting question. And then of course, it has this lovely evolutionary puzzle to it, which is that reductions in survival early in life prior to reproduction, we think should have the highest fitness consequences. And so the big question I've been interested in is, well, why don't we see the evolution of juvenile resistance? Shouldn't the plants evolve their way out of this issue? And so when I started this, the first thing I thought was like, well, let's actually explore that first question and ask, is it actually selectively advantageous to have more resistance as a juvenile? So we asked this question, when is juvenile resistance favored? And to do this, I built this really simple, again, just an SI model. And you've got two classes, adults and juveniles, and they can both get infected at different transmission rates. And the juveniles mature into adults at a set rate. So we just have one little period of juvenile, and then they emerge, and only the adults are reproduced, right? Babies don't reproduce. And so and then we looked at three. This is the first sample model. We looked at three genotypes of this fungus, and or of this pathogen. The host. And we said, well, one could be fully susceptible. So this is showing you the age along the bottom. There's a little chunk for juvenile, and it's the adult stage. And so, okay, and then this is a proportion infection. So the susceptible one is just susceptible the whole time. There's a juvenile onset resistance that's resistant the whole time, and that resistance onsets first thing. And then there's this adult onset resistance, which is what we see in dianthus and a lot of these things, where it's susceptible at first during the juvenile period, and then a switch is hit, and it like turns on its resistance later in life. So we wanted to know basically how do these evolve in a natural population. And so what we did was we had this model, and then we can just start with a population that's fully susceptible and introduce these two genes and see what happens if they are spread in nature. And we assume cost. So we assumed that resistance is costly, and it takes a fecundity cost. And we assumed that this juvenile onset resistance is a little bit more expensive than the adult onset resistance because it's more resistance. So this is what we find. I'm going to just cut straight to the results here. So you can see here's how we ran the simulation. Time here, this is the frequency of each genotype. So we start with a fully susceptible population in a population that's stable at 40% resistance, because this is what we see in these natural populations of anther smut. And when you introduce these two genes, so the adult resistance is in blue and the purple is in juvenile, initially they both increase in frequency because it's better to be resistant than to be susceptible when there's this much disease. And in fact, the juvenile onset resistance is favored over the adult resistance early on in the epidemic, or early on in the evolution. But then actually it changes, right? And you end up losing juvenile resistance entirely. And you sort of end up with this fixed stable polymorphism between adult onset resistance and susceptibility. So why does this happen? Well, the other thing that you can see here is that basically there's a feedback. So as resistance evolves, you can't maintain the same amount of disease in the population. So the prevalence of disease starts to decline. So as the prevalence of disease is declining, the probability of getting infected decreases, and the average age of first encounter increases, right? So there's less disease, so the time at which you might expect to get that first encounter increases. And so if it increases to the point where you basically don't expect to get it as a juvenile, then there's no point in maintaining any kind of resistance, even if it's quite cheap. And so basically what we think is that there's a feedback happening where evolution of adult resistance is changing the ecology, and it's actually maintaining seedling susceptibility through this ecological feedback. So I've been working now with Ben Ashby. He's a real theoretician and has been doing this with adaptive dynamics models. So this is the kind of thing where you can actually, instead of just setting resistance at particular levels, you can actually evolve it quantitatively using these sort of trade-off surfaces. And so he and his grad student have been doing a bunch of this work to basically do this, but allow adult and juvenile resistance to evolve independently according to trade-off models. And one of the things they've been testing that I think is really interesting is what kinds of trade-offs generate this, right? So is it any kind of a trade-off? And so they've been looking at trade-offs between resistance and mortality, maturation rate. So maybe you have higher resistance, but you have a slower maturation rate, and then just adult fecundity. And I'll just show you, they just put this paper out, and I'll show you one result. And basically what they've found here is that if both forms of resistance carry equal fecundity, we see adult resistance evolves to a higher level than juvenile resistance. And so here, what they're doing, this is the level of resistance. So this is the dashed line, is sort of the evolutionarily stable point that comes out in these models. And they said this also depends on the degree of sterility caused by infection. So diseases that are more virulent or more sterilizing are going to generate stronger selection for resistance. And that if the fecundity costs are equivalent, that you actually get adult resistance always evolving to a higher level than seedling resistance. So they see the same feedback. And you can see it's also happening here because, again, we're changing the total amount of infected hosts. So as adult resistance is evolving, you're getting less disease, and that feedback is what's driving us. And so I'll return to this graph in a minute, but keep this result in the back of your head here. Okay, so far, what I'm hoping I've convinced you is that evolution of adult resistance can actually help maintain juvenile susceptibility through ecological feedback. So we don't necessarily need to invoke like a constraint model. I was sort of rapidly taking slides on my talk last night because I was worried about making it too long, but we have actually found that in Dianthus, there's a lot of genetic variation for resistance at the seedling stage. So we know they can, and we can select them in the lab. So we know they can evolve higher levels of resistance. We don't see it in nature. So we think we can explain this partially just through ecological feedbacks. And basically, we've also shown, or Ben and Lydia have shown, that juvenile resistance doesn't necessarily have to be more costly than adult resistance. So we're not really talking about like the cost of juvenile resistance is so high, and that's why we see it. Okay, so but then I'm not happy to just stay on theory. So I want to know, what about real life, and what about smut in particular? And so we had some questions just about the evolutionary potential of age-specific resistance in this system. And we want to know, first of all, are these even separate traits? You know, in all the models, we just assume there's like a gene for juvenile resistance and a gene for adult resistance. Is that real? Or is it just the same trait with a constraint on it, right? And then this question of costs, like how do costs scale with age? So most studies of resistance costs are just good at one age, and we don't have an idea actually how they scale with age and if costs are stronger in particular. So this is work we've been doing now in my lab, and for most of this work, we've actually switched systems, and instead of working on the Dianthus system, which is beautiful and has great ecology, the plants we can't get them to flower more than like once a year, and they're really long-lived, and they're really hard to affect as adults. And so we've switched to working on this other system, Semini-latifolia. So it's got healthy flowers, healthy males and females. It's got two sexes, and then but it gets an anther smut. And this is a related species of Microbacterium, but anther smut's a species complex. So there's lots of these things. They're all in the sort of broader Caryophyllaceae carnation family, and they each host plant has its own smut species. So there's not a lot of like cross affection between them. But the advantage of this guy is that it's fast growing. Its lifespan is about 2 to 3 years. And it's a European species, and it's not European, but it's been introduced to the US in the 1800s, and it's up and down the eastern seaboard, and the smut is here also. And so we can grow it in the lab without permits, and we can actually do field experiments in Maryland and have the fungus out there too without getting in trouble. And unfortunately, I decided to work on this. I just started at Maryland, and then the pandemic hit, so it wasn't like I was going to go to Italy anyway. So it was really good that we collected all these CITES plants and had plans to do some local stuff. So that would be another tip: is like, even if you think you don't need a local system, you never know when a pandemic is going to hit. So our approach here has basically been, we've generated, and I should acknowledge right here that I'm going to show you a bunch of work, and I absolutely have to credit our technician, Ali Keto, and postdoc Sam Flonsky, who've done just a tremendous amount of work rearing these plants and then trying to rank all the data. So we started with a panel of 45 full SIV families. These were collected from a wild population in Virginia. First thing we did was we did some resistance phenotyping. So we inoculated these at we reared these to 4 different ages in the greenhouse, spanning from about 1 to 4 months. And we inoculated them and scored them for infection. And one of the important things about working on this SMUT system is you can't score infection. An individual plant is either infected, it's not infected. And so if you want to know how susceptible a family is, you have to grow up like 100 seeds and get a proportion. And so we do one experiment called a greenhouse, because we're like, each one of our data points is like 100 plants. So so the sample size looks impressive, but the data that I'll show you will look less impressive because but just remember how much work went into each one. Okay, and then we did a field at the same time. Actually, we started the field before because we got shut down in the greenhouse. But we took those same plant families and we planted them out in a farm field, and just in a common garden experiment. We had 16 replicants of each family, and we just wanted to measure their fitness in the absence of disease. And so we do basically under field conditions. What's fitness in the absence of disease, and how does that correlate? Are there evidence of costs of resistance, and how do these costs scale across ages? So that's the plan. And so we'll talk about the first question first. Can resistance evolve independently at different ages? And so for this one, what we really wanted to know is, are these separate traits? And so one thing we thought was, if there's a really strong correlation between infection rate as a seedling and infection rate as an adult, it might be sort of the same trait, and it could be pretty constrained, such that if you evolve resistance at the adult stage, you're going to drag along resistance to the juvenile stage, and then that would kind of suck because a lot of our models would like fall apart. So that was one possibility. The other possibility would be if there's really no correlation. If in fact you can be resistant as an adult but susceptible as a seedling, or vice versa, then maybe this is really there's a lot of evolutionary potential to evolve these as separate traits. And so we took this correlation approach to do this. And so here's what we found, and I'll show you first. So here's the resistance phenotyping data from the greenhouse. So first, what I'm showing you, this is just age. Everything, the y-axis is always going to be proportion infected here. So this is age, and so you can see we've got, again, in semi-neonatal, we have a clear age pattern, but at least we can infect the adults here, and we're not dealing with 4% infection rates. So the seedlings, especially the really young seedlings, they're just a little less than 1 month or 3 weeks old, they get infected at a much higher rate. But interestingly, now in this graph, this is showing juvenile infection along the x-axis and adult infection rate along the y-axis, and each one of these circles is an individual plant family, with the size representing how many individuals we actually were able to score in that family. And there's just no correlation. And if you look at the age by family interaction, it's all over the map. So that most things are more resistant as adults than as seedlings, but we have a few families that go in the other direction, and the slopes are just all over the place. So there really doesn't seem to be very much correlation at all between these two forms of resistance, which we think is interesting. So we've decided, yeah, there's no correlation here. So we think these are separate traits. And we've actually done this in 2 other species as well, and found similar things. So in Dianthus as well, we find really strong age by family interactions. So it seems to be mostly true across it, except we have one species of Silene vulgaris where we actually do see really strong correlations in age. So it's not universal across all of these different plant species, but fortunately, the one we've been putting a lot of effort into, there's nothing there. So then we wanted to ask, okay, well, how do costs of resistance scale with age? And so this is where we were testing for a cost of resistance with this field. This is our common garden in Beltsville, and I had no idea how big Silene plants get if you irrigate them and put them in a field and take care of them. this high. And our plan was to count flowers, and we ended up having to like quickly abandon that. We took photos, and then we were like going on a log scale and walking down the story. And so they had a really good time. They did a lot of flowering. What we were thinking here again is that basically, so I'll show you some graphs in a minute, but our thought here was that if we found a really steep slope, so infection rate, this would be things that have a high infection rate are going to be more susceptible, things that have a low infection rate are going to be more resistant, and this would correlate with fitness. So if you see it, even though I'm talking about trade-offs, if you see a line going this way, that would be evidence of a trade-off because things that are more resistant have a lower fitness or flowering rate. And over here, we thought that basically, okay, if we can find differences in these slopes, then it might mean that a really steep slope should have a fairly strong trade-off for cost of resistance, and maybe a weaker slope would have a weak cost. And so one hypothesis would be, maybe it's just that seedlings, one of the reasons we find high seedling susceptibility could just be that seedling resistance is really costly, and maybe it's just dirt cheap to have it up resistance, and so it doesn't matter. So this is what we've been looking at. Okay, and then I have some more polished data, but I'm a big fan of showing raw data, actually. So this one's, so here's our actual raw data, and this is what you're seeing. This is male plants, so we had to break these plants. Some of the plants are half of them are female, half of them are male. So we looked at, and males make a lot more flowers. So we'll look at males first, and each one of these graphs is showing infection rate. So this way is like more susceptible. This is more resistant. So exactly the same for the graph. And this is total flowers in the second year. And this is at different ages. So this is 9 days, 19 days, all the way up to 104 days. And so what we found actually is that, and in fact, all of these lines turn out to be significant. Even this one that's irritating and going the other direction. And we find similar things with females, but it's less significant. So these ones are not. So, but in most cases, it seems like there is a trade-off between susceptibility as measured in the greenhouse and flower number in the field. And it's actually, we see it across ages. And of course, I've cherry-picked one piece of fitness data to show you guys. This is total number of flowers in the second year. But we collected a lot of data on these guys. So we also looked at survival rate, and we looked at flowering in the first year, and sort of timing of flowering. And so what we did was we actually used these aster. Oh, that's weird. Okay, the error bar is not showing up on this, but I figured instead it's actually a little cleaner to see it. So what we did was we used this aster model that is a way to integrate different components of fitness, particularly survival and cavity components. So this is actually a 2-year estimate of fitness that includes both survival and flowering estimates for all of these plants. And these are their slopes, and each have sort of imagine some error around them. But what we found is that basically we find significant costs of resistance in all ages, except for this one here. And this is like not even the oldest age. This is the second oldest age, and we have no idea what's going on there, other than we had a really hard time infecting plants in that one. So we're just missing a lot of, we think that maybe the inoculation didn't go well or something. We're actually repeating these 2 ages right now with a higher inoculation dose to try and increase infection. So we can see if that's actually a real fit. I suspect it's not, but maybe if you hear me talk about this in 4 months from now and have the data, I'll know for sure. But anyways, we think we see pretty strong, we're definitely getting significant slopes in all of these, except for this one here. And so this change in flower number is actually related to plant fitness. So it's a real fitness cost, not just one trait. And so then we thought, well, we'll compare the slopes across these different ages. And so here, this is age of inoculation, so you're getting older over there, and then this is the slope of the susceptibility, right? And so what you can see here is that there's basically no change in the slope. So there's a consistent cost of resistance across age, but it's consistent. It's not like the seedlings have a way higher, like a steeper trade-off with fitness than adult resistance. And of course, this one's negative in the wrong direction. So, one of the things I want to point out, though, is that actually, we had a big debate about this in the lab conversation. Because our hypothesis, and I was like, well, no, because remember, actually, that when you have fecundity costs, having actually consistent costs that are similar at both adult and juvenile resistance, we still would actually predict that this should favor the evolution of higher levels of resistance in adults and lower levels of resistance in the seedling, which is exactly sort of what we're seeing. What we'd like to do now is actually take these actual real trade-offs that we've measured and go back to Ben and Lydia and see if they can like put in, instead of fictional numbers, like real actual kind of plant numbers to round this out. So is resistance costly? Yeah, we're clearly seeing real trade-offs with fecundity, and these are related to fitness. But importantly, costs are actually consistent across ages. So there's no evidence that seedling resistance is any more costly than adult resistance. And of course, Ashwini's theory predicts that similar cost of resistance should still drive the evolution of higher adult resistance. So I'm going to just sort of wrap up here by giving you a visual model for how I think this is happening in the system here, which is the dynamics of persistence are really occurring both numerically and through evolutionary change. And so imagine you've got a population of plants consisting of adults and seedlings, and a pathogen comes into this population. What we think happens is initially there are two modes of transmission. There might be pollinator transmission, which we expect to be highly efficient because the spores are being picked up and deposited on an actual host. And then this less efficient aerial transmission, scattering spores and hoping to hit something. But what we think happens here is that when you have this initial transmission occurring, this elicits really strong selection for adult resistance that rapidly sort of makes this transmission no longer very good for the pathogen. And consequently, transmission shrinks to adults, and also it reduces the pathogen population size. And as that pathogen population size decreases, it's small enough that the probabilistic chance of getting infected, but as a seedling decreases. And so you actually have sort of, even though it's very bad as an individual if you get infected, overall selection for susceptibility is quite weak, or selection for resistance at the seedling stage is quite weak. And so you hit this sort of valley where you can't get out. And so the disease is able to persist because the seedlings are still susceptible, but the seedlings aren't able to evolve resistance because the disease isn't prevalent enough to really drive it all the way there. And so the thing that really is sort of getting me excited now is thinking about this, how does coevolution work here, right? What do we expect? How do we expect the pathogen to respond to this, right? You could imagine that maybe there's just a take it like prey on the weak, specialize on the seedlings because they're already so susceptible, and take advantage here, and maybe just forget about trying to infect these adults. Or maybe there's selection for increased, directly sort of fighting off this adult resistance, because maybe you get a bigger bang for your buck if you can infect adults. Then you can use this much more efficient transmission route. And so I don't have an answer here. This is like a question that we're excited about and interested in. I'm actually, since I have 2 minutes, I put 2 slides right at the end that I will, and I'll say is we are working on this now. And I have 2 really good undergrads in the lab that just asked this question: do pathogens actually vary in response to juvenile and adult resistance? So this is new data that isn't quite, we haven't even quite finished counting it, but I want to show it. So what they did here was they did a cross-inoculation experiment with 5 strains of the pathogen against 4 strains of the host, but at 2 ages, seedlings and adults. And these were all actually not our Virginia strains, but these were all from the native population in Europe, because we really wanted to tap into areas where there'd be a lot of genetic diversity. And they wanted to ask basically, are there pathogens that are specialized in adult hosts? And so this is what they found so far. And so here, this is showing you host age, 2 weeks, seedlings, 8 weeks, adults, and then these lines are different pathogen families. And what you can see is there's significant pathogen by age interaction. So the one that's most infectious on the seedling stage is actually the least infectious on the adult stage, but then this other guy, the FX7, that's pretty consistent across the whole thing. So we think this is pretty exciting. The other thing is it also varies with host genotypes. So if we look at all 4 host genotypes that we tested, there's actually a 3-way interaction, which is kind of annoying because I can't even quite wrap my head around it. I wish there wasn't actually. But what we're seeing is basically that there's this. You can see that there's sort of a strong age interaction in almost all the families. This one actually has, sort of susceptible across the board, but that the pathogens are interacting differently with age. And so we're really excited to start thinking about how is this actually working in a natural context. So I will just go back then to my conclusion slide and say, I hope what you've taken away from this is that resistance evolution is just totally essential for host persistence. We can't think about this as an ecological problem. It's definitely an eco problem. And that resistance doesn't occur evenly across the host lifespan. Juvenile susceptibility is essential for disease persistence, and I think it's maintained by these ecological feedbacks and costs. And I hope that also convinced you, even if you study it and said that can be pretty cool. So that I would say thank you to a bunch of people, all the transmission work, especially Giannis and Hanovics, Michael Hood, and our 2 Italian collaborators. And then I've had a lot of help with the age-specific resistance, and Ashley, Lydia Buckingham, and then Sam Swincy, Ali Kiddo, and Andrea Sheridan have done really a lot of the heavy lifting in the field, and the rest of it. Thank you.
So if your day 78 data holds up, like, you know, you resample and it holds up, maybe this is a question for Ben Ackley, but does that actually change any of your conclusions in terms of whether there truly is a trade-off or not? And like,
It might, yeah.
If you're 78 days out, does that change the implication in any way?
Yeah, this one here, where we've got the 70. Anyway, it could. I will tell you one more piece of biology that I didn't mention, which is that it's not just age. There's also a stage component here. So adults can vary in whether or not they are, the plant sort of grows as what we call a rosette, so vegetatively, and then when it's going to flower, it sends up a spike, so then it's bolting or flowering. And so actually, at the 78 and the 104 days, some of the plants had already started to flower before the experiment started. Much more of them were already in flowering stage at 104 days, and there was some variance in 78 days. And we swizzled, so we marked which ones they are. It turns out that plants actually are even more resistant when they're in the flowering stage. So if you compare adults within the same age group that are still vegetative, they're much more susceptible, and when they start flowering, they become more resistant. This is kind of cool. It's like backwards from what we think of in a lot of animal systems, where when you switch to reproduction, you actually like there's a trade-off there, and so you're investing in reproduction with less resources for defense. But the plants actually seem to be jacking up their defense when they're going into reproduction. We think it could have evolved because as they start to flower, this is when they're going to be exposed to this smut fungus, and we've actually tweaked it with like using gibberellic acids and stuff to like induce flowering in plant and light treatments and stuff, and it holds up pretty consistently. But we still can't. I say that, and I still can't explain this entirely by using that. Otherwise, I would have put it in. So we actually just don't know what's going on there, and that's what we're just following down and repeating it.
So, Mike, can you go back to the slide with the theoretical results? See that one of those.
This one or the previous
Yeah,
one?
that one's fine.
Yeah.
So here, they had equal competitive cost, right? So I'm just thinking back to kind of the figure you had where, so juvenile resistance kind of comes on during the juvenile stage, but then kind of persists throughout the whole lifespan, right?
Yeah.
An adult one just kind of comes later and then persists, right?
Yeah.
And so if those have equal costs, you know, I still don't understand why adult resistance would evolve, but not so much kind of juvenile resistance, because even if you actually have a lower level of infection, right, the force of infection is kind of constant at a lower level. Still, the distribution of kind of age of infection. I mean, it's the average age of infection is higher, but it's still exponentially distributed. So you're still going to get juveniles infected, right? So I'm just wondering why, if it's equal cost, right? Why not get that extra little boost of kind of being protected kind of earlier?
Yeah, that's a good question. And when I was doing it in my simpler model, I had to actually have a slightly higher cost of juvenile susceptibility in order to maintain it, but it was almost negligible. So if it was just a little bit more, it would do it. And here, they're both. So in this case, and I think what's happening is what they're actually doing is like they're running these simulations where they've sort of separated, I hate the terminology, but ecological and evolutionary climb, and so they're allowing like mutations to occur, and then those mutations have to be able to invade a population. And so I think what happens is as you get up into this stage where you have adult resistance, that invasion potential is dropping here. I would say I've also cherry picked this, and there are other results when they were looking at these other trade-offs, mortality and maturation rate. Actually, you get different outcomes. So with a mortality trade-off, you actually get sort of similar levels of resistance involving at both, or actually higher juvenile resistance. And with maturation trade-offs, it's also again more it favors juvenile resistance. So it's really just the fecundity trade-off that seems to be driving. And the reason they actually set them the same, so they're different overall because this is like the rate of fecundity, but then in each case, the juveniles themselves are not actually reproducing.
So, when thinking about the like aerial transmission to juveniles of the little like spores, does wind speed impact like how far they like disperse, or is it pretty set? Like,
I have two answers to that. One is we don't quite know the mechanisms of how they transmit. So we've done some things where we've put them out. We've set up flowers and then put spore traps out in different directions to see if like wind speed matters, and we don't see any obvious variation. It also doesn't seem to matter if there's like healthy flowers above them. So we also thought that like maybe pollinators like sort of cleaning their legs next to plants, but we find no evidence of that. And we also, I spent a couple of weeks doing these goofy experiments where we were like taping flowers and trying to build a wind tunnel, and like first we used IDAS's hair dryer, and then we brought in an industrial fan. And we couldn't get the spores to fall off of the plants. So they were just like sort of stuck on. There could be some splash dispersal. So when you spread water, they definitely spread. And then there's also there's an age component with the transmission too. So as the disease flower gets older, it gets more crumbly and falls. But it's actually a bit aggravating. We would really like some fungal traits that control like sort of how sticky they are. Because then we could really track, how does, can the fungus evolve to be more
Right.
And I should say that we know that that if you look at the whole phylogeny of anther smuts, the basal microbotriums are all aerial transmission. So the production of spores in the anthers is a derived trait. So we think that vector transmission by pollinators is a derived trait. And we found at least one example of something that's sort of reverted back. So in Neotides, it's this tiny little flower that's like visited by flies. And when that gets infected, the spores are produced in the anthers, but they kind of blow up the whole flower. And there's just a big black clot of spores everywhere. And no pollinators are visiting it. So it seems like something that can evolve, but we don't have a good, we haven't been able to find a good measurable trait yet. We've messed around with like stickiness, and we're trying to do other sorts of like quantifying, and we've been totally unsuccessful with that.
I have a question about pollinators, and I guess maybe this is because I don't know anything about plants. But like the diseased plants they're not producing pollen or seeds. Are they producing like nectar?
Yes.
Okay, so there's no difference for the pollinators.
There is a difference.
Okay.
So we've actually measured this. They do produce less nectar than healthy plants. And we've actually quantified, because people ask this in every seminar. And so finally, I've been sure this. So I had some really good undergrads that did a honors project on this. And so it depends on the pollinator. So some pollinators actually strongly discriminate against diseased plants, and those would be hoverflies and bees, and it tends to be mostly things that are pollen gathering, and they will avoid diseased plants. They will also avoid female plants. They just go for the hermaphrodites. But things like butterflies tend to not really care, and they're going for nectar, and they'll just hit all the time sort of equally. And so we still sort of don't know. We've done a lot of pollinator transmissions, and we still don't quite know which pollinators are actually doing the like heavy lifting in the system. Because you can definitely see these hoverflies come along and they'll visit a whole bunch of little flowers in one area, but the butterflies come, they just have a much larger, like they'll 1, 2, 3, and then they'll go much further. And there's a whole group of like little teeny flies that just sort of hangs out.
Yeah, I guess I was just wondering like how,
But there are real pollinator preferences, and they tend to avoid, overall, they tend to avoid the disease. Although I've seen some bees that like, there seem to be some bees that are like, they must be trained on the disease flowers, and you can see them trap lining just the diseased ones. And if we were trying to like fish for them, we were putting out healthy flowers, it seemed to get them to crawl into a healthy flower right after diseased flowers, so we could see, well, how many spores do you get from the direct transmission? And it was really hard to get the bees to do that if they were the ones that were actually on the disease. I think they'd avoid, like, develop a sense for.
thanks.